Hi everyone, how are you? So are you ready for learning today, this morning? Let me share you the slide first. Huh? Okay, here it is. So this morning we're going to learn about DNA as genetic material. The objective is for us to understand why DNA is called as a genetic material, okay? It is a basic actually before I continue to the next chapters related to the DNA replication and also protein synthesis. Okay, so let's try to understand it together, yeah? Okay, before we start further, when you hear about DNA as genetic material, what do you think about it? Or what do you know about it? You can tell me by telling me directly, but you can also tell me via chat. Okay, let me check the chat box here. Okay, just tell me whatever you think about DNA as genetic material. Ah, okay. Here, Ferdinand says it's connected to hereditary. Yeah, okay, good. That's a good point. Anybody wants to tell something? Oh, okay. Irene here, huh? Irene says because the NA is inherited. Yeah, it's connected. Okay, so what Ferdinand and Irene say are correct actually. And now we're gonna see further. Yeah, here it is. So actually for a material to be called as genetic material, it's it should fulfill these three requirements. Yeah, the first one, it must be able to store genetic information. Remember? To store genetic information. Yeah, that's the first requirement. The second one, it must be able to be replicated accurately and it should be stable. Yeah, so the second requirement is it should be stable and able to be replicated accurately. Okay, especially during cell division, yeah, and because we know that DNA is located in the nucleus of our cells, yeah, and then it can be transmitted from generation to generation, like what Ferdinand and Irene said, yeah, and the third one, it must be able to undergo mutations to provide genetic variability. That's why we can see many variations, for example, in the color of our hair and the color of our skin, and then the height of our body, the color of the flower, and everything else, okay? Because this third requirement should be fulfilled. So remember, okay? Three requirements for a material to be called as a genetic material. Yeah, store genetic information, stable and able to be replicated accurately, and able to undergo mutations. So DNA can fulfill all these requirements. Yeah. So what's the proof actually? Okay, let's see the proof. Here was the first proof from the early experiment. Yeah, we can also say it's actually one of the history of the DNA invention as genetic material. Yeah, Griffith's transformation experiment. So at that moment, Griffith injected mice with two strains of pneumococcus. Okay, the first one was the encapsulated one or S strain, and the second one was the non-encapsulated one or R strain. The S strain is virulent, so the mice would die. Okay, and when the sample was taken from the mice, it has a mucus capsule and forms shiny colonies. And the R strain actually is not virulent, so the mice still alive. And from the sample, it was known that the colony has no capsule and it was dull, not shiny. Yeah, so you can see from the picture here. The first picture showed you that the mouse died after it was injected with the S strain that was virulent. And then the second picture here, the B picture, the mice still alive, yeah, when it was injected with the live R strain, okay, because R strain was not virulent. And then you can see here the picture, the third picture, the C picture. The mouse was injected, the mice was injected or were injected with the heat-killed S strain. Yeah, so the S strain was killed by the heat first, and then it was injected to the mice, and the mice are still alive. And now we'll see the the fourth picture here. So the mice were injected with the heat-killed S strain plus live R strain. Yeah, so both of them, okay? The heat-killed S strain and the live R strain. Yeah, and the mice died. Okay, and when the sample was taken from the mice or from the mouse blood, for example, it was found that the S strain was was discovered. Okay, you can see here a live S strain is withdrawn from the dead mice. So when the sample was taken, Griffith found the live S strain. So what happens here actually? You can see here, yeah, especially in the fourth picture. First, the mice were injected with the heat-killed S strain plus R strain. But then, when the sample was taken, it was found that the live S strain was there. So what happened? Okay, that's the first proof, and we're gonna see the second proof first. So after that, we can conclude. Okay, this was the second proof conducted by Avery, McLeod, and McCarty. Okay, so they used actually 3 enzymes. Oh yeah, before I continue, I usually speak a little bit fast. So if you think that I speak too fast, you can just interrupt me, tell me, or you can also chat or tell me in the chat box. Okay? Yeah, I'll try to slow down a little bit. So these 3 scientists used 3 enzymes: DNAs, RNAs, protease, yeah, to digest substance, okay, which allowed Streptococcus to produce a capsule and become virulent. Yeah, we can see here from the picture. So they use the heat-killed S cells. Yeah, remember S cells is actually the virulent strain, yeah. And then here, in the first step, the scientists remove the lipids and carbohydrates from a solution. Okay, so the remain in the tube are only proteins, RNA, and DNA. And after that, the scientists groups or divide the tube or the treatments into 3 treatments. Yeah, 3 treatment groups. The first one here, they add proteinase. Okay, and the second one here, they add ribonuclease. And the third here, they add deoxyribonuclease. Now we'll see what happens after that. On the first tube here, yeah, they took sample and they found that there's no protein in it. On the second tube here, no RNA, yeah, remember, okay? So it's ribonuclease. We're gonna see again the difference actually between RNA and DNA, just to remind you, because I believe that when you learned biology in your high school, you knew about it already, yeah? So the second tube here contains no RNA because they use ribonuclease enzymes that digest RNA, okay? And on the third tube here, the sample contains no DNA because the enzymes that degrade DNA was added. And after that, what happened next? Yeah. The next one is actually they add a small portion of each sample to culture containing R cells. Okay. And then they observe whether transformation has occurred by testing for the presence of virulent S cells. So as you can see here, they add R cells into each tube. Yeah, on the first tube here, after they add R cells and they took samples afterwards, the S cells appears here. Also the second tube here, yeah, they added R cells and then they took samples and the S cells appear. So they said that transformation occurs on these tubes, on these two groups. And then on the third treatment group, they also added R cells, but no S cells appear, so no transformation. Okay, so they concluded actually that transformation cannot occur unless DNA is present. Therefore, they tried to conclude it further. DNA must be the hereditary material. Yeah, okay. That's the second proof. And now we're gonna see the third one, conducted by Hershey and Chase. Okay, they used a virus called T-phage that was radioactively labeled. Yeah, the DNA of the virus was radioactively labeled. Also, they code proteins. Okay, in separate experiments, and they used E. coli here in these experiments. Okay, now we'll see also from the picture. There are two bacteriophage treatment groups here. Yeah, bacteriophage is the name of the virus. Okay, to be more specific. Now we'll see here the left one. The sulfur, yeah, in the protein capsule of the virus, it was labeled, yeah, like radioactively labeled. So it's kind of molecular label that allow you to track, yeah, to track the compound in the cells. Okay, we're gonna see the second one here is the DNA phosphorus. Okay, the DNA of the virus here, yeah, was labeled. Yeah, phosphorus labeled DNA core. Okay, so the phosphorus here on the DNA was labeled. And now they allow the virus to infect the E. coli, yeah, on the both treatment groups. And then after that, the virus, of course, will, like inject the material genetics into, or the DNA into the E. coli cells to form new viruses structures. Okay, so the second one is blending, and after that they centrifuge. So centrifugation is actually they took the samples, they put it in the solution in the tube, and then they stir it in the machine. You will know later. Okay, the machine is called centrifugation, centrifuge. Okay, they spin it. Okay, by the gravitation. So the cells are, so yeah, we can see here, yeah. Yeah, the cells of the E. coli will sediment because it's spins by the gravitation. And on the first group here, the sulfur was detected in the supernatant. Yeah, in the supernatant means, so after you did the centrifugation, all the solid materials that was heavy will go down, will settle as sediment, and above it there will be a liquid. Yeah, so that liquid is called supernatant. Yeah, so on the first group here, no sulfur detected in cells. Yeah, it means in cells is solid, so it's sediment. But the sulfur was detected in the supernatant. That's on the first group. And on the second group, the phosphorus was detected in the cells. Okay, and no phosphorus detected in the supernatant. So that's the difference. Okay, it means that radioactively labeled DNA, yeah, on the second group, as you see, but not protein, ended up inside bacterial cells, causing them to be transformed. Yeah, that's why we can say here, they can say, and now we can say that only DNA was the genetic material, but not protein. Okay, so that's the third proof. Yeah, the third proof. Sorry, that's the third proof. So Are there any questions related to this? Are there any questions? Oh, okay. So who is this? Let me see. Alberto, okay. Alberto was asking about, oh, the previous one. Yes, this one. What does it mean by mucus capsule? Oh, okay. So I will try to explain it a little bit again. Yeah, the difference between the S strain and the R strain. The S strain is virulent. The R strain is not virulent. You know, the virulent bacteria usually they have a capability to form a capsule, a mucus capsule. It's actually a structure that was formed in an unfavorable environment. For example, when the bacteria was injected to the mice, okay, so the immune system of the mice will try to fight it, of course, and that environment become unfavorable for the bacteria. So in that kind of situation, the S strain, the virulent one, can form a structure, a protective structure that is called capsule. Okay, so it will be more resistant to the immune system of the mice. That's why the mice died when they were injected with the S strain. Yeah, so you will learn about this more later in the maybe in the second or third year. I think it's second year, of course. Yeah, second year in microbiology. Okay. Yeah. So is it clear enough? Okay. Now we can continue. Yeah. Now let's see about the structure, DNA structure. You can see here, yeah, the picture DNA here. And DNA, as you know, is actually an abbreviation of deoxyribose nucleic acid. It is, of course, a nucleic acid. And now we're gonna see one terminology, a nucleotide. I think you have already heard about it, but I think maybe some of you have already forgotten what nucleotide is. So now we're gonna try to recall it, yeah? One nucleotide or a nucleotide is a basic building block of nucleic acid. So nucleotide here is a basic building block of DNA. Yeah, because now we talk about DNA, not other nucleic acids. Because nucleic acids are not only DNA. And a nucleotide is composed of one pentose sugar. Pentose, don't forget, pentose means 5 carbons. So the sugar contains or has 5 carbons. Yeah, so a nucleotide is composed of one pentose sugar, one phosphate group, and one nitrogenous base. Okay, remember that? A nitrogenous base. Yeah, so these three structures compose a nucleotide. You can imagine a nucleotide actually as Lego, so you won't forget. Okay, now let's see, yeah? So there is one pentose sugar here, yeah, has 5 carbons. It is attached to one phosphate group, and then it is also attached to one nitrogenous base. We're gonna see after this what is nitrogenous base, yeah? And these three structures in the end form one nucleotide. Okay, just remember this picture as Lego so you won't forget about nucleotide. Guys, okay guys, yeah. We continue first. So this is the structure of DNA. Yes, I just tell you about, the previous one, nucleotide. So one nucleotide will connect or will attach or will bind to another nucleotide. So one nucleotide attached to another nucleotide, and these two will attach to other nucleotide. You know, it's like a chain, yeah, and become DNA, okay? So how can one nucleotide attach to the other? It was actually bound by a chemical bond, phosphodiester bond here. Phosphodiester bond, yeah? It's a bond between adjacent nucleotides. And this bond is actually formed between the 5-phosphate group of one nucleotide and the 3-hydroxyl group, or OH. of the next nucleotide. As you can see here from the picture here, these are the first nucleotide, and here the second nucleotide. Okay, now we're gonna see here, yeah, the phosphodiester group, the phosphodiester bond here, yeah, this is the bond, is actually the bond that formed between, yeah, between the phosphate group here on the C number 5, sorry, this one, yeah. So this is the first nucleotide, and we can actually we can give the names, or not calculate, count the carbons in the sugar. Okay, uh, let's see together. This is the first carbon here attached to the base, and this is the second carbon, this is the third carbon, yeah, attached to the hydroxyl group, or OH. This is the fourth carbon, and this is the fifth carbon that is attached to the phosphate group, yeah. And this fifth carbon that is attached to the phosphate group form a phosphodiester bond with the third carbon here on the second nucleotide, or adjacent nucleotide. Yeah, that's why here DNA, or nucleic acid, has actually a kind of direction or orientation. Yeah, the chain of nucleotides has 5 prime. We call it prime. Okay, this apostrophe things, we call it prime, has a 5 prime to 3 prime orientation. Yeah, remember that. Okay, because this is the basic for the next chapter about replication and protein synthesis. Okay, now we're gonna see about the next structure related to nitrogen base. Okay, now we see. Remember in your high school, or you also learned about it, that there are several nitrogen bases: adenine, guanine, thymine, cytosine. Okay, and Chargaff was another scientist that explored or studied about DNA, found actually that the composition of this one, okay, you can see here. You can see the tables, yeah. So there are a lot of spaces here, and there were compositions of each nitrogenous base. You can see here from the same spaces. For example, the first one, Homo sapiens, or human. You can see that the composition of adenine here is equal to the composition of thymine. And the composition of guanine here is equal to the composition of cytosine. Yeah, and now we can also see the structure here. This is adenine. Okay, this is adenine. And then this is guanine. So they have 2 ring structures like this, yeah. And then this is thymine. Okay, thymine. And cytosine has only 1 ring structures. Yeah, don't forget about that. Later you can see again the picture, of course. Now let's come back to the table. Okay, so now you can see here in the same spaces, adenine and thymine composition is equal, and then guanine and cytosine compositions are also equal. So what can you conclude about it? Let's check from another species. For example, here, Escherichia coli, the bacterium, huh? The composition of adenine here is equal to the composition of thymine, and the composition of the guanine here is equal to the composition of cytosine. Okay, so finally, Chargaff concluded something, that in each species, okay, in the same species, there are equal amounts of adenine and thymine, and then equal amounts of guanine and cytosine. Yeah. So from this experiment, he also concluded that DNA used complementary base pairing to store genetic information. Remember, yeah, that was the first requirements of the genetic material, to store genetic information. And DNA stored the genetic information in the nitrogenous bases actually. Okay, and those nitrogenous bases complementary, yeah, complementary to each other, but only A and T and G and C. That's why you learn in your high school that the adenine is complementary to thymine, and guanine is complementary to cytosine. Okay, this was the proof from Shargav's experiment. Okay, and then related to the structure of DNA, another scientist that was famous, Rosalind Franklin, also actually found the structure of DNA using the X-ray diffraction. And it was confirmed by Watson and Crick. Okay, you know also about Watson and Crick? Yeah, I guess you heard about them already. Because these two scientists actually the one that make famous, okay, about the model of DNA as double helix. So DNA is double helix, okay, and then it has sugar phosphate bonds, okay, in the helix, and the hydrogen bond actually will connect or will make the complementary bases attached to each other. And then the two strands of DNA are anti-parallel to each other. Okay, anti-parallel means it has a different orientation. You remember the orientation is related to the carbons, this one? Yeah, the information stored in DNA must be read in the 5' to 3' direction. So DNA is replicated in 5 to 3 direction. But you can save this information for later, for the next chapter that I'm going to teach you about the DNA replication. Okay, just remember that DNA is double helix. It has sugar phosphate backbones, and then the hydrogen bond attach the complementary bases. And then the two strands of DNA are anti-parallel to each other. Okay? As you can see in the picture, here's the model of DNA proposed by Watson and Crick, the model that we used until now, yeah? You can see here, DNA is double helix like this. And then these are the backbones, okay? The backbones of the DNA that actually sugar phosphate backbones. Here the sugar, the phosphate, the sugar, the phosphate, yeah? So it forms like a backbone for the DNA. And then you can see here, okay, remember that one nucleotide composed of one sugar, one phosphate, and one nitrogenous base. And you can see here, there are two complementary nucleotides. Why I call it as complementary nucleotides? Because there are complementary bases here. Like this one, guanine and cytosine, and here thymine and adenine. Yeah, and the complementary bases are connected, okay, or are bound with hydrogen bonds. Okay, remember, yeah? The nucleotides will be bound to another nucleotides by phosphodiester bond. Okay, so phosphodiester bond form the backbone of the DNA. But the complementary nitrogen bases are bound together with the hydrogen bonds. Yeah, and the DNA or the chains of the DNA are anti-parallel to each other. You can see here the orientation. This one is 5 to 3 and the other one is 3 to 5. That is called anti-parallel. Yeah? Okay, so any questions? Any questions about this, guys? You can just ask me anything. Okay. Let me check. Maybe if there is a question in the chat box. Oh, okay, it's Ivan. Ivan, you asked about, Miss, can you explain again about the anti-parallel things? Yeah, of course, yeah. So remember that, maybe I will show you this picture first, just to remind you. No, not this one. This one, okay? Phosphodiester bond. Yeah, phosphodiester bond is a chemical bond that connects one nucleotide to the other one. Yeah, or one nucleotide to the adjacent nucleotide. Okay, and how is the bond is formed? So you can see here from the picture, the bond is actually formed from the carbon number 3 of the, or we can see just here, sorry, yeah, this one. From the carbon number 5, okay, maybe I should put this one here and this one there. Yeah. Wait a minute. I will try to fix it first. Sorry, guys. Yes. Yeah, I think it's better. Okay. Let me just save it first, and now let's continue. Yeah. Okay. This one. So this is the phosphodiester bond that is formed between the carbon number 5 here, yeah, on these nucleotides, and the carbon number 3 here on the adjacent nucleotide. Okay, and how is this how it is calculated? You can see here from this sugar, pentose sugar. This one is sorry, I should use the laser pointer. This one is the first carbon. And here is the second carbon, here is the third carbon attached to the OH group, and here is the fourth, and this one is the fifth carbon attached to the phosphate group. Okay, so because the pattern is like that, that's why we called, yeah, that the DNA is actually has orientation. We call it 5 to 3. This is carbon number 5 to the carbon number 3, 5 to 3. Okay, that's the orientation or the direction. And the DNA is double helix. Okay, it's double helix like this. So you can imagine it as how to say, it's maybe like what is tali in English? Rope. Yeah, like two ropes. Okay, there are two ropes, and those two ropes, okay, have orientations or directions that is anti-parallel to each other, like this one. You can see it from the picture, number C picture. The first rope, okay, here, has orientation from 5 to 3, yeah, prime. We call it prime, okay? And the second one has orientation from 3 to 5. Or we can also say from 5 to 3, because that is the correct orientation. But now the left one, 5 to 3 from up to down, yeah, in the picture. And the right one, 5 to 3, but from the bottom to up. Okay, that's why we call it anti-parallel. Is that clear enough, Ivan? Yes? Okay, thank you. Okay, so if there's no more question, I have a question for you, just to remind you before we finish. So what do you think, why I can call DNA as a genetic material? What are the requirements of the genetic material? Raise your hand. Okay, Alberto, yes. Well, what is the answers? You can chat it. You can also type it in the chat box. Yes, correct. The first one, it should what? It should actually, it can be replicated, yes. Yeah, it should be stable and can be replicated, okay, accurately. And then what else? Yes, it can store genetic informations, and we learned about DNA just now. The genetic information are stored in nitrogenous bases, okay? And then what else? There are 3 requirements. What is the third? The third requirements. You forget about the third? But well done, Alberto. You can mention 2 requirements. So who can help Alberto to mention the third requirements? Okay. Who? Oh, yeah. Alicia. Okay, Alicia, what is the third? Anti-parallel to each other? Okay, nice try, but that is not the answer. Okay, just to remind you once again, let's see, yeah. Here it is. Okay, so the requirements for the genetic material, it should be able to store genetic information like nitrogenous bases in the DNA, and then it should be stable and able to be replicated accurately. And the third one, don't forget, yeah, it must be able to undergo mutations. We're gonna learn also about mutations later in the next chapters after you learn about DNA replication and protein synthesis. Okay, I think that's all for today, guys. So prepare yourself for the next chapter. I have already uploaded some materials for you to read a little bit, okay, before we start the lecture. Keep calm, yeah, because practice makes perfect. Okay, see you in the next lecture. You can also ask questions outside of this hour if you want, okay? You can just contact me. Bye, everybody.